presentation. We come now to the next speaker, who is Cristianne Rijcken from Cristal Therapeutics in Maastricht, and she will talk about the CriPec platform and the lead product, CriPec Docetaxel principles, preparation, biomedical applications, and clinical translation.
Thank you for the kind introduction, and I would like to thank the organizers for being capable of presenting actually an update about the clinical progress of CriPec Docetaxel. But maybe first get started. Cristal Therapeutics, we are a company, as indicated, in Maastricht located. We are originally a spin-off of Utrecht University, where actually I did my PhD work under supervision of Professor Wim Henning, and that was back in 2007. And now in 2018, I'm very proud that we can present a couple of clinical trials and also some very interesting progress towards phase 2 trial. So what we develop is that we developed this so-called CriPec-based platform, nanoparticles, of which I'll depict the underlying principle in one of the next slides. But it's a broadly applicable platform technology able to entrap both small molecules, bigger compounds such as peptides, but also oligonucleotides, and can be used for different applications. We develop products ourselves, as well as also in collaboration with other pharmaceutical companies, and we do that under a venture capital-based structure, so we are a private company. So the underlying principle, and I don't need to explain the EPR to this public, but the underlying principle of CriPec is that we envision that nanoparticles, assuming they're long enough circulating as an intact nanoparticle in the bloodstream, they do accumulate in either the tumor tissue or in other organs, and that only selectively in these organs the entrapped compound is being released. So overall the aim is as we all aim, increasing the efficacy while also at the same time improving the safety profile, so really shifting the therapeutic index. And the approach that we have taken is that we make nanoparticles in a different strategy as than, for example, the more traditional liposomes. Namely that we have a drug, we derivatize it with a linker molecule, so really a covalent bond is being generated, and then if we combine it with our proprietary polymer, the mixture self-assembles it to initially a micellar structure, but then thereafter is immediately cross-linked. So I like to stress that one nanoparticle is actually one big macromolecule in which both the drug molecules as well as the polymer chains are all covalently linked to each other. And then, if preferred, we can conjugate to the surface also a targeted moiety or any other compound. But as you can imagine, such a nanoparticle has a different biodistribution over the body, as indicated by the concept in the former slide. But what we really aim is that over time, only the native drug is being released as a result of chemical hydrolysis. So that means that by using different type of linkers, we have full control over the exact release kinetics. While at the same time the nanoparticle also disintegrates into smaller fragments and will also ultimately be cleared by the kidneys. So what we've done in recent years is we actually proved the broad applicability and tunability of the platform. So not only that we were capable of making very small up to quite larger nanoparticles, while the larger part is not the really attractive part, but particularly going to small particle size with the entrapment of different type of modalities and even entrapping 2 APIs in one single nanoparticle, each with its own release kinetics. And not unimportantly equal is that, well, you can make all kind of materials small scale on the lab, but ultimately it's about the clinical translation. So we were very fortunate to have been capable of actually upscaling towards clinical scale in a very straightforward manner. So no complicated equipment required, but just standard organic chemistry equipment. And by that, we've now produced quite a lot of clinical batches successfully. So this platform, as I said, broadly applicable for either existing compounds, that what we do ourselves with CriPec Docetaxel, but also for novel compounds that were formerly not feasible. So we have the ambition to also develop a CriPec oligo product ourselves. And in collaboration with partners, we also work outside the oncology arena, also in the immune arena, as well as in other indications. But due to time reasons, I cannot disclose that at the moment. So let's jump to the results obtained with CriPec Docetaxel. So very snapshot of all the preclinical work done. This is a study where we did an equimolar comparison in a breast cancer model. We only gave one single administration of either Taxotere, so the current commercial product, or our product. And I think the picture speaks for itself, namely that after one single administration, we really have a long-term tumor growth suppression. all as a result of, and that was done in a parallel study, of a much higher tumor uptake of the CriPec Docetaxel in the tumor tissue. So in a separate study, we demonstrated we get up to 40-fold higher uptake in this orthotopic model. But what was even more important for us as well, not only the total Docetaxel, so that's representative for the intact nanoparticles, but in the tumor tissue there was a sustained release of the Docetaxel, so the native API, and that explains to us a really long-term suppression of the tumor growth, that there is a continuous exposure to released API. So then I'll make a very quick jump, because I think last year I showed you the underlying preclinical studies, the GLP studies. This is the Napoly trial, which was the first phase 1 study, the dose escalating approach, where in the PK parameters we're determining, I show you only 2 dose cohorts here, where we compare it versus Taxotere, as well as 2 former competitors called Bind and Cerulean, former because both of the companies don't exist anymore. But still the data are very good as a reference. And particularly I'd like to point your attention towards the half-life of the nanoparticles, because that's what we're looking here at. This is so-called total Docetaxel. The half-life of the nanoparticles in the clinical situation is around 33 hours, really indicative of this long circulation. While in case of, for example, Bind, they have a much lower half-life. And also another parameter, what you can take a look at, is the AUC, so the actual exposure in the bloodstream. That is also significantly higher for us as compared to cerulean compound. So that was already promising from a PK perspective. But then if we, actually the last patient just went off study, and we're now compiling all the data. So this is not the full final answer, we're just gathering the clinical study report at the moment. But this is the conclusion that we can draw so far, that the unique profile has also been demonstrated in the clinical setting. And whereas Taxotere has a main dose-limiting toxicity of neutropenia, actually there was hardly any neutropenia observed in our phase 1 study. That's mainly from a clinician perspective a really major benefit, but from a patient perspective, the absence of alopecia is also a very attractive point of CriPec Docetaxel. And in fact, what we also dare to deduce is that the really changed distribution of the adverse event profile gave us also evidence for an increased and actually changed biodistribution profile also in a clinical setting, in combination with some encouraging signs of target response. It's a phase 1, so it's not a lot of statistics there to be done. But ultimately we came to the conclusion that the recommended phase 2 dosing is 60 milligram per square meter every 3 weeks. So that's what we're going to go forward with. But in fact, next to this Napoly dose escalation study, we're doing 2 separate studies, phase 1 studies in parallel, studies that really focus on towards not only proving the safety as well as the PK, but particularly proving the enhanced tumor uptake also in the clinical setting. And I think that's where the field is really lacking the majority of the data. And I'm very pleased that we have set up this study together with Professor Matthijssen from the Erasmus University in Rotterdam. I think this is a study that is unique worldwide. Namely, what we do in this setting is that we treat patients either with Taxotere or with CriPec Docetaxel, and then the other way around. So actually, each patient is its own control, and then we take biopsies from liver metastases. So a patient is treated, then liver metastasis is being taken and treated with the other regimen, and then liver biopsies again taken. And what we do, we take those biopsies at different time slots or time points, as we envision that particularly at the longer time stage we do see a differentiation at the tumor uptake. The study is still ongoing, so it's not yet finalized, but the data that I can show today are very, I would say, supporting and promising, namely that Taxotere, it's the blue bar, you see an uptake which is significantly less as the total Docetaxel, so the nanoparticles as such. I dare to say a roughly 4-fold higher uptake of the nanoparticles in the clinical setting, and where we see also that the released Docetaxel coming from this nanoparticle is well, roughly the same as compared to Taxotere, but please be aware that this is measured up to 3 days. So the initial differentiation maybe initially there is not a lot of a differentiation, but particularly upon the later time points, so we will measure also 4 days, 7 days, and 14 days. There is we really envision to see the bigger differentiation, similarly as we've seen in the preclinical study. So this is work that's still ongoing. But as it's obvious, liver biopsies are not really patient-friendly approach. So what we've done in parallel is that we also set up an approach to monitor the nanoparticles non-invasively. And even more importantly, this is a study that is, or a full program that's supported by a Horizon grant. What we do here is we conjugate zirconium to the surface of these nanoparticles with the ultimate aim, so this is like we're now only doing the first steps, the ultimate aim to achieve higher uptake in tumor in patients and only selectively treat those patients and the patients who do not show this increased uptake to not proceed them with CriPec Docetaxel. So what we've demonstrated is we've been capable of producing these nanoparticles, but then we had to demonstrate also that this modification does not affect the PK and biodistribution profile. And without going into details, in this case, we administered the plain particles as well as the modified particles. And we see actually a same blood profile as well as similar tumor uptake in tumor-bearing mice. So that that allowed us to conclude that actually the bioequivalence is not affected by this modification. So that it really also enabled us to go forward into the clinical translation. So that's the summary so far is that we have a very good PK profile observed, the really prolonged exposure, also increased tumor uptake with also an increased safety profile. And well, what we are currently working on is the 2 phase 1 studies have to be finalized. So the CriTec study as well, the Piccolo, which is the non-invasive imaging. First patients have been treated, but the results are to be processed as we speak. And then the most exciting part is, of course, getting started with the phase 2 study, which we've called CNOVA, as we're going to evaluate CriPec in ovarian cancer. And for that, we have now started up in 4 countries, applications in 10 sites. So it's really exciting to see how the ovarian cancer patient is going to be responding to CriPec Docetaxel treatment. And then I very briefly shift gears to another follow-up product that we like to develop ourselves, which is CriPec oligonucleotides. And I think there are a lot of challenges in the oligonucleotide area, but the main hurdle is, of course, bringing the oligo there where it's supposed to work. Namely, in our case, we aim to tumor targeting. Well, I've shown you already for some compounds, the stability in the blood as well, as that we also have higher tumor uptake. But what we foresee application of the CriPec is that we can change both the internalization into cells, the local release of the compound from the nanoparticle in the endosomes, and then also ultimately the endosomal escape. But at the same time, what we're also working on is to make sure that the siRNA itself is sufficiently optimized, meaning that it's stable enough under the conditions that it may encounter in the endosomal escape as well as upon traveling into the cytosol. So in terms of the increased tumor uptake, these are data that are already slightly older. We've done it already in the past, showing the increased uptake upon active targeting, selectively in this case of RGD, but there are a couple of other ligands possible. And only in case of the integrin positive cell line, we do see really increased uptake. We do have already demonstrated that we have selective release of, in this case, an siRNA that was entrapped under slightly acidic conditions by using different type of linkers. And then a last step that is even more attractive to show, so most recent data, is that this is only a showcase where we entrap the same siRNA into nanoparticles. If you inject it into mice as a naked siRNA, it's, as known, rapidly cleared. But in case if we entrap it into CriPec nanoparticles, either plain or RGD targeted, then you really have this long circulating in the bloodstream, which ultimately translate into a very good tumor uptake over time. And as said, if the siRNA is not in the bloodstream, it cannot reach the tumor site. So this is for us really an example, long circulation, also good uptake into the tumor tissue. And with that, we're currently working on that. This is what we're doing in-house as some sort of a high throughput screening. We're currently trying to tick all the different boxes that are essential, as I indicated in the concept, and working there to go forward to measure anti-tumor efficacy of a model and actually also potential product siRNA ourselves. So with that, I hope to have given you insight into the application of CriPec, as well as also the clinical translation of the CriPec Docetaxel. And thanks for all your the patients for their participation and also for listening. I'm happy to answer any questions.
Yeah, thank you very much for this presentation. Are there some questions? Yes.
Cristianne? Just one question. For your phase 2, how come, was there reasoning behind choosing ovarian cancer?
Yes, there was clearly a reason. It was a well thought, evaluation. And ultimately it comes down that in ovarian cancer there is still a high medical need for the platinum-resistant patients as well as it also, the Doxil has a proven efficacy there. So nanoparticles have been used in this set of patients. So the combination made us to conclude to go forward with the CriPec Docetaxel in ovarian cancer.
There's one more question.
A very nice presentation, and really nice to hear that the success you have of your product. I have a question about taking out metastasis sample that often. The reason I'm asking is that we have a discussion in Trondheim, and then the oncologist said that no, we could not take out any liver metastasis, just measuring them by CT.
Right. This is a center of excellence for these type of metastases. So actually, the radiologist does take these liver biopsies, particularly of the liver metastases and not of the healthy tissue. So that's the only answer I can give. That in the Netherlands, it is possible to do so.
Are they in the back, Danny?
Could you comment about the polydispersity of your particles?
Yes, absolutely. So what we've produced, either small scale or big scale, the polydispersity is always less than 0.2.
Okay. Oh, there's one more question, the last one.
Thank you very much. I was a little surprised by the phase 2 recommended dose being 60 milligrams per square meter. Mm-hmm. I guess that's even below free drug docetaxel. And it's slightly against the current belief that you could maybe increase the dose, maybe you can even lower it and gain more efficacy would be great, no?
Right. Well, I would say the dose that is given in patient is variable between 60 and 100 milligrams per square meter. So there is some variability depending also on the indication as well as the patient population. And ultimately it comes down to what is the adverse event profile that you see, but also how much of the drug can you bring to the tumor. So based on the CriPec data, we think that the combination of the phase 1 safety data, the pharmacokinetics, but particularly the CriPec data, we've shown a higher uptake. So you want to be on the how to say, the optimal side of efficacy versus accumulation, that you thereby expect highest efficacy.
Okay, then thank you very much for this presentation.